Our next speaker is Dr. Flavell, who is also a member of the National Academy and of the Institute of Medicine, and who is our neighbor at Yale. And he is the chair of the Department of Immunobiology. I don't know why the other day I saw he had more than 1,000 papers. So interestingly, he has been in the academic world for a while, probably a few decades, but before that he was a chief scientific officer of Biogen. And at the time that Biogen was formed, when Charles Weissman cloned alpha interferon gene, which has been, of course, an important thing. So now Richard said that he would talk about humanized mice, but he just told me that he has changed his mind and he would talk about something else. He would talk about, guess what? Microbiome. But I've asked him to add a few humanized mouse cells. I don't know whether he will do it. Richard does what he wants. Richard.
Well, thanks, Jacques. It's a great pleasure to be here. And first of all, my congratulations to you all on this great adventure. And welcome to Connecticut. It's a great place to do research. I've been here longer than anywhere in my life. I came here in '88, and I'm still here as of today. Let's hopefully tomorrow it will be still true. And it's just a fabulous place. And as Jacques says, actually, we're hoping very much to have collaborative interactions, and Jacques and ourselves are very interested in humanized mice, and that will, of course, feature in the story I'm going to tell. And the story, the reason I chose this topic is it's really about the intersection of your genotype and the environment, and that's really an essential part of what we understand about human disease. And the focus of this talk will actually be human disease itself. So we, of course, you've heard a wonderful talk already today on the human microbiota. We have trillions of these organisms living inside us at every surface at which they can reach, and they can reach a lot. And they outnumber us in an enormous way. And the concept really that we became interested in over the years is the fact that that there would be many of us and many people in the audience, most people in the audience have, quote, a healthy compendium of organisms in our various places. This is, of course, focusing on the intestine. But that in some human disease, as a consequence of genotype and environment, that this compendium of organisms would be altered, and there would be, let's call them bad bugs, present here, and those bad bugs are a contributor to the disease which this person is afflicted with. And the complexity of this microbiota, of course, has been a subject of a lot of analysis, but I think most people are thinking in humans that there's an excess of 100 species and probably more microorganisms that are living within us, but of course each of us has a somewhat different collection of organisms. Now, of course, as an immunologist, I can remind you what you already heard earlier today, and that is that we have sensing systems in our genomes, and these sensing systems are ancient. They go back to early evolved species like arthropods, early deuterosomes, all kinds of organisms. And these are systems, in fact, essentially, if you're a multicellular organism, you have this kind of sensing mechanisms, and they are designed to determine whether you are exposed either to microorganisms of various classes, or they will determine the consequences of that exposure when it leads to damage to our tissues. And you've heard about toll-like receptors, RNA detecting systems, DNA detecting systems, and a variety of sensors of tissue damage. Again, damage to cells that's mediated commonly through microorganisms, but also through environmental insults of various kinds. And so these systems are, of course, have been subject to enormous evolutionary pressure. And what they do conceptually is they survey the environment around us, around each of our cells, either the outside, inside vesicles, or in the cytoplasm, because, of course, there's trouble coming from everywhere every day of your life. Now, our focus in several years has been on one particular member of these families, but all of them are of importance and interest, and they could take hours and hours to discuss them. But I will talk to you about these NLRs because they will lead us into a discussion of environment, microbiota, and genotype. And this NLR family, these are, again, ancient and complex gene families. Things like sea anemones have hundreds of genes of this type. They're, again, involved in pathogen microorganism recognition. They're very similar to molecules in plants, which play the same kind of role. We have focused on them as potentially playing a role in microbial recognition and damage recognition in mammals, in humans, mice, etc. Now, one of the most interesting things that came out of this is, in fact, that if you look at members of this family, and if you take one particular member of this, and there's no point in going into detail, but just imagine one of these things. Here, this is one particular kind of NLR, which is, contains these modules. And some of these are really capable of integrating all kinds of inputs, but the basic common denominator is cellular damage, as I mentioned. And so if a cell receives some kind of damage signal, that is integrated into a very simple output. And one example of that would be just to take this molecule. This is really work from Jörg Chopp, who first established this point. But the whole point here, again, the details don't matter, but the point is there's an input coming from a variety of different either microorganisms, uric acid, product of metabolism, all kinds of things. And the output of all of those different things, and there's potentially hundreds of inputs, is extremely simple. It's the activation of a protease, which is caspase 1 or caspase 11, and the output is the cleavage of substrates which yields cytokines. The cytokines then send, these are of course messengers which tell the immune system that there's trouble, and they will then drive an inflammatory response. The second output is an output of cell death, and that leads to, of course, to a protective mechanism whereby you can eliminate the trouble-causing cell plus its new friends, the pathogens that have just joined it. And so the basic principle of this is view these systems as amplifiers of complex inputs and telling our bodies really what's going on and how to interpret that. Now, downstream, as I said, of this is inflammation, and inflammation, we now know, is one of the biggest contributors to chronic human disease that we're aware of. So going back to this point, you have to imagine that if you imagine you have these sensing systems, then the place where you've got to do a lot of sensing is, of course, where your natural friends, the microorganisms, are, because they're with us every day of our lives. Salmonella is with us now and again. It's not very nice when it is, but these guys are with us for the whole time. And so it occurred to us that there would have to be some sensing systems that would tell the organism, us or the mouse, how to maintain a homeostatic relationship with this collection of organisms. And so that's the question. Did these inflammasomes, that's what led us off to studying this, and we published the first paper on this back in 2011. Do they actually modulate this thing, and are they involved in this homeostasis? And of course, when you do that, you have to do an experiment. So the experiments, again, I'm not going to go into a lot of experimental detail for you today, but the experiments were to ask the question, if we perturb these inflammasomes, and of course one perturbs them genetically by using gene targeting in animals, does that then lead to a change in the homeostatic relationship and, in other words, a different intestinal microbiome? And long story short, that was in fact the case. And what we did, starting with one of the members of one of these pathways, we picked this gene, took knockout mice with it, and asked, if you don't have a functional gene of this kind, can you regulate your microflora appropriately? And the answer is you can't. And so once we found that, we went searching for the other members of the pathway. And of course, then we analyzed this, and this. These are the ones that gave the positive outcome, and many others which did not, of course. And this turned out to define a pathway, which I'll show you how it works in just a minute, which actually plays this regulatory process. How did we get there? Well, I'll just show you one silly experiment. This is just a way of measuring the healthiness of the relationship between your intestinal microorganisms and your intestines themselves. Now, we all know that there are inflammatory bowel diseases, Crohn's disease, ulcerative colitis. These are serious problems, and they're in part caused by this non-homeostatic relationship because of the genotype of the individual concerned. I want to look at the microbiome in that context. And what you can see here is that basically very simple outcomes. This is how sick you are. The lower this goes, the sicker you are. And you give mice a perturbation. And this is a wild-type mouse, and this is a mutant. You see billions of experiments of this nature. And sure enough, the mutant's sicker than the wild-type. Well, at that point, you say, who cares? We have got another gene that makes a mouse sick. But that wasn't the interesting point. The interesting point was, if you took these two mice and you actually put them in the same cage, then in this case, they're both equally sick. In other words, what we have is a sick mouse, this mutant mouse, which is actually able to transmit its disease susceptibility to this mouse as long as they live together. So it's a cohabitation or environmentally driven susceptibility to disease. So then we went to work that out. As I showed you, we found out the pathway that was involved. And of course, this transmission suggested it's microbial. That's the first thing you would consider. And bottom line is that they all phenocopied this susceptibility, as I just mentioned. And it led us to a model whereby in the epithelium of the gut, because that's where this gene in particular is expressed, it's selective for that location. This pathway is functioning there, and it's keeping the environment of the gut, and I'll show you the evidence for that in just a tiny second. It's keeping our gut environment sterile, essentially, or superficially sterile, keeping the bad organisms out. And if it doesn't work, then you get what's called dysbiosis. You get changes in the composition of the microbes of the organism which has this deficiency, invasion of the intestines, triggering inflammation, which is what this is showing here. And once inflammation occurs, you have the key hallmarks of this inflammatory bowel disease, of course, in a mouse model. Now, we were saying there had to be microorganisms involved. Was there something selective, or was it just random? So we went looking for the microbes, and I'll just spend a second on that. And sure enough, when you do this, you find microbes. And what we found is there were some microorganisms highly selectively associated with this, particular gram-negative anaerobes of a family called Prevotalaceae. Again, don't worry about that too much. And they really are intimately associated with the gut. This is an EM picture, and this is a crypt. You remember the gut has this epithelium there, these crypts where the stem cells are at the bottom, and they make new cells. Here is this is what's called goblet cells. That they are making mucus, which gives a layer of material, mucus, which keeps out the organism, and keeps them away from driving inflammation under normal circumstances. But if you don't have the pathway I just described, this is what you get instead. You get invasion of the intestinal environment, so the crypts. You get hyperplasia of these goblet cells, the mucus-driving cells, and you get disease. And so this is the reason that this this under normal circumstances is not working. What you have is actually, and that we looked here, in this environment, it's not just every organism is present. Selectively, there are what we call bad bugs that are getting in here and causing trouble. Now, we actually now know why that is, and I'm just going to show one slide for that. This is because, as I just told you, this is the epithelium here. These are the microorganisms living outside, and normally there's this layer of mucus I just mentioned to you, and this mucus layer keeps out bad bugs to a degree, together with lots of other defense mechanisms, of course. And what we now found out, and I'm going to skip the details on this, is this pathway that I just described to you actually functions in these cells, may function in more places, but at least in these cells. And actually the production of this mucus is controlled by this pathway. And so we actually are using the sensing system for microorganisms to titrate how much of this protective layer you actually need in order to keep them out. And that makes a lot of sense, of course, because you'd like to make this in a way that matches the need. And this is the evidence that shows that's true, because this is the mucus layer in a normal mouse, and in the mutant mice, the mucus layer is almost absent. It's very defective because the material, the mucus, cannot get out of the cell, actually. It's a secretory problem, and I'll go into more detail about that, but that's basically the mechanism which is underlying the deficiency in this system. So you have a sensing system that's involved in making this barrier. And that's the basic idea. So you imagine organisms get through here, they trigger an antimicrobial, the sensing system. The system says, okay, make more mucus, and then you keep it going like that. So back to this system. Well, that's all very nice. What it told us is that you can have in animals organisms that cause disease, that they are being detected and so on. What about humans? We were able to identify these microbes to a degree in the animals because of a lot of selective and hard experiments and hard work. But how do we figure out what's going on in people, where it's a lot more complex? And now a lot of beautiful work's been done with human microbiota in causally correlating the organisms with diseases. But what we'd like to do is, of course, establish a cause and effect relationship and understand how this is all going on. And so the hypothesis that we had, of course, come up with is there really are disease-driving members of the intestinal microbiota. And in the mouse system I've just shown you, this is one of them. But of course, there are others well characterized. Helicobacter, a famous microorganism which is involved in humans in gastric problems, and others. And these are the concept is these are disease-driving members of the microbiota. So how do we produce Koch's postulates and identify these organisms? I should point out at this juncture that the dysbiosis that we showed in that previous study, we went on to show is actually extremely pleiotropic in its disease-driving potential. And those mice, which I showed you get IBD, are susceptible to all of the manifestations of a metabolic syndrome we looked at. So they become obese because of those microbes. And that's the driver. If you get rid of the microbes, they don't become obese. They're the same as their non-dysbiotic littermates. They become type 2 diabetics. They develop fatty liver disease. These are all manifestations of metabolic syndrome. They also develop colon cancer, a double to 3 times the rate in certain models. We've only done a couple of models, but big effects in these models. And so this is likely, this issue, this bad bug issue, is of course likely to have a lot of impact in many, many manifestations. But we wanted to translate this situation to the human. And so the question was how to go about that. And so what we postulated is that if you have this situation where you have pathogenic microorganisms, be they real pathogens like salmonella and so on, they are, of course, brilliant at getting through the barriers, and they drive inflammation. We then make an immune response to them, and we clear them through that immune response. Now, you would imagine that most of the commensals, the ones that we're friends with, have a symbiotic relationship with us. and they would in general be in a homeostatic relationship, just as I mentioned in the beginning. But these organisms I was just talking about, the ones that actually are living with people in a chronic relationship, rather than pathogens, which are usually in an acute relationship, that they would drive chronic inflammation, that would drive disease, but it also, you might expect, might drive immunity, because that's inflammation that does drive immunity. And so we postulated that if that's the case, then these organisms that are in these two classes, the ones that are so intimately associated with us, they will actually drive immunity. And we all know, and many of you have learned in college, that immunoglobulin A, or IgA, is the antibody we make, which is designed to deal with these kinds of problems. You make more IgA than any immunoglobulin. You make 2 to 5 grams a day. It's been associated with the microbiota. It's involved in neutralizing toxins, all kinds of functions. And you make it because once the organism gets to the gut, you will sample that through your immune system, and as they say, shit happens, and eventually you make antibodies, and out they come, and they deal with the bugs. So we speculated that if that's true, then the bugs that are a problem have a special property, which is they drive this process, and they therefore ought to have antibodies bound to them of this class. And so that was the idea. In other words, that the commensals would be only perhaps have low affinity antibodies bound to them, but the pathogens and the inflammatory microorganisms that live with us, they will have high affinity antibodies. And so if that's the case, then we could use this. First of all, we could prove the point, but secondly, we could identify the bad bugs. And so this is the hypothesis. You've got the disease-driving members. They drive inflammation. They drive antibody production, but we can use them to identify it as well as to try and do something about it. And so this is the idea. And all these diseases I mentioned to you, metabolic syndrome, IBD, of course, colon cancer, as you know, are inflammation-associated diseases. So this is what we did. And again, who cares about the details? Well, what you do is you collect feces, you grind them up, you use a secondary reagent to detect the IgA, which is bound to the bugs, right? It's bound to the bugs still in the feces, because that's what happens. It's released, and then it floats downstream with the bugs. And you just use a separation technique. So we use cell sorting. And this is an example. This is bugs which are going through a fax machine. These are bound to an immunoglobulin. These ones are not, or are bound only to a low level. And then you sequence them. And then you know what you got. And in theory, if we're right, these are bad bugs and these are okay. Well, to test that first, of course, we used a known. So we took our dysbiosis system, and the way that we did this is, remember, these are the names of those mutant mice. Again, who cares? But remember, we had a wild-type mouse, which is healthy. We have a mutant mouse, which has colitis. And we have a mutant mouse that makes a wild-type mouse colitic if you put him in the same cage. So we just compared the microorganisms, these guys, and this is what you get. It's a series of bugs, and this is the phenotype that I just showed you earlier. What about the coating with the antibodies? Well, this is the coating. This is a coating index. It's the ratio of the presence of the organism. These are a bunch of organisms. Are they found mostly in the IgA high class or the IgA negative class, all right? And the more they're bound to, the more of them that are in the antibody-bound class, the bigger the number. So it's really not complicated. You get 3 or 4 things in this mouse that are actually, you make a lot of antibodies to, and they are the ones that we would be concerned about. This one is the one I showed you the pictures of, the one that's found in those crypts, the Prevotalaceae. They're highly coated. Here's Helicobacter, highly coated. SFB, highly coated. And this is the actual numbers. Who cares about the numbers? This told us that this was really working, and therefore it would allow us to move forward. Now, we did a few controls, obviously. This high IgA coating is mostly dependent on a T-cell response. That's because it's a strong T-cell dependent response. So that fitted with the concept that we had in the hypothesis. And then, of course, we needed to do the important stuff, which is to go to humans. And we could have taken any of these diseases, and we shall move forward with several more of these. But again, we had the same concept that in patients with colitis, there should be, or inflammatory bowel disease, there should be disease-driving members, and we ought to be able to use this approach to find out who they are. And so we did this system. We got fecal material from healthy humans, humans with Crohn's disease and humans with ulcerative colitis, the two main classes of inflammatory bowel disease. And we just looked to ask, if you go down this list of microorganisms, how many of them are coated and how many are not? Which ones are coated? Now, color coding is coating. The yellow is highly coated. Gray is a little hard to see. I realize, and I apologize. Gray is almost not coated at all, and if it's black, they're not even there. This is what you find. This is in individuals, and so you can see a few things immediately, which is a given person does not have many highly coated bacteria, and that's just like those mice. Also, if you're a Crohn's disease patient, you have organisms which are not found in healthy controls, which are coated. You see the same, and you can look at more details of that. Obviously, this is not about detail, but you could classify organisms as to only found in UC, only coated in UC patients, only coated in Crohn's patients, coated in both of them, or only in healthy, etc. Okay? So interesting correlations, but of course that is correlation. And here you see it in a bit more detail. So the question is, how do you take this correlation to causality? And remember that this is our hypothesis that the IBD-associated IgA-inducing bacteria are the ones that are selectively driving pathology, and we want to identify them because then we can do something about them. And so if that is true, then we ought to be able to take the organisms out of the patients and culture the ones which are highly IgA-coated. And this is what we did. We took 11 IBD patients, grow them up, and sequence, of course, to know which ones we have. And then we made personalized IBD strain collections, which we could then use to do causality experimentation. And so what we did for the first round of experiments is to make consortia of bacteria which really don't elicit an IgA response. You can see here, it's all gray or very pale blue, okay? So these are ones we picked of the ones these are all derived from patients, by the way, but these are non-coated. And these ones are all coated in somebody. Remember that individual people are not the same. Someone has this one coated, someone has that one coated, and so on. But we made a mixture of these organisms, and we then put them in germ-free mice. And so now these germ-free mice have human microorganisms as a consortium, those which don't have antibodies bound to them in a person, or those which do have antibodies bound to them in a person. Well, they grow quite nicely in mice? They slightly redistribute. This is the mixture that went in. This is how they settle down in the mouse, but it's pretty reliable. And they do what in mouse what they do in people. This is a couple of weeks after they've been in there. They tell the germ-free mouse to make antibodies. And so this is the IgA positive consortium. They tell them to make more antibodies than the IgA negative consortium. So they kind of phenocopy what happens in the human, as you can see here. Well, okay, so then, but what about the effects on disease? Well, if you grow up the mice under normal circumstances, at least for a couple of weeks, the mice are actually okay. They don't get sick. But if you then perturb the intestine in exactly the same way that we did in that first set of experiments with the mouse, then all hell breaks loose. And what you see is actually, and this is disgusting mouse guts, slide number 1. So this is what happens if you get IgA negative consortium. This may not look very nice to you, but actually this is a happy mouse intestine. These are feces, and this is a normal mouse. The colon you measure the length as an assay for how healthy it is. If you get the IgA negative consortium, these mice are perfectly healthy. But if you give them the IgA positive consortium, they are really, really sick. And these mice, basically by few days after this, they're at death's door almost. Their colon length is contracted. They're bleeding in the intestines. They have the worst pathology that our pathologist who did everything blind has ever seen in a mouse study on IBD. And these are human human bacteria which are coated with IgA in patients. And this is what you can see here. This is a complete mess, basically. Whereas the ones that get the IgA negatives have normal histology. So this tells us that these bugs are actually pathogenic in a germ-free mouse, so that we can transfer their hypothesized hypothesized causality in these diseases in humans to at least a direct causal association in a mouse. Now, of course, in a mouse, it's not in a human, but it's a pretty good start to a description of what's going on. Now, just to finish up now, there's still a couple of interesting things. And one of the things was that bacteria from the same species, when you look at them, actually display differential coating in different patients. And you can see this, because if you go across here, you see this is gray. That bug is there, actually, in that patient. But in this patient, it's coated. So why is that? Is it the host? I haven't gone into this, but of course, which I should have. But of course, you all know that Crohn's patients have a very well described set of particular alleles of immune genes, many immune genes, genes involved in autophagy, genes involved in a whole series of things. Is that part of the issue? Or is it the environment of the host? The hosts eat things. Our environment involves food, right? The microbiota's environment involves food. Or is it the genetics of the microorganism? And so we purified a couple of these guys. One that's highly coated from this particular patient. Its ICI score is almost 40. It's as high as anything that we've seen in the mouse studies. And the other one, which is from the so-called B. fragilis, same named organism, it's essentially non-coated at all. And then we sequenced these guys. And sure enough, they're actually very distinct strains. That may not be an enormous surprise, obviously. Neither of them, by the way, are identical either to the type strain which is used, which is considered to be immunoprotective. And this is the one that's highly coated. This is the one that isn't. And this is just lining up so you can see difference visually. So what about, are these single organisms different in their pathogenicity? And the answer is yes, they are. The one that's highly coated, if you put just that in the mouse, it makes the mouse sick. And you can see here, this is looking at the colon length. Again, this is the one that's coated. It's called the same thing as the other one. And the one that isn't, when it goes into, these are now monocolonized animals, doesn't cause a pathology. And so, or I shouldn't say doesn't, causes less pathology, of course. These are relatively simple assays that we perform. And so it suggests that there's, of course, much more complexity, and that underlying complexity is that of the organisms that are present in different people, some of those are of different levels of pathogenicity, but the immune system actually can tell the difference in this. Now, last point, what is it about these IgA positive, quote, bacteria, in other words, those that elicit an immune response, that causes this pathogenicity or immunogenicity? And what we find is, and maybe I'll just use the next slide, which is a higher power. What we find is it goes back to the thing I started right in the beginning, which is the organisms that are in this IgA positive consortium actually can get access to the gut epithelium. They can get through the mucus layer. So this is in a mouse, which is a wild type mouse, remember. It's a germ-free mouse, but it was a germ-free mouse. This is the intestine wall. This is just DNA staining. This is the microbiota, which is normally kept outside. But in this case, in the IgA positive consortium, these bacteria can get into this mucus layer and therefore can drive the inflammatory response, which we characterized earlier. That's not true for the IgA negative consortium. They're kept out by the so-called demilitarized zone, which Laura Hooper, a nice phrase, coined for this. And so this is probably a fundamental difference, which we think is perhaps causally involved in this pathogenicity and immunogenicity. So just to finish, so we think that, very simplistically said, health and disease as far as colitis is concerned is very much influenced by the composition of the microorganisms that individuals have, in some cases, organisms that make them susceptible to disease. Of course, the genotype of the individual is crucial in determining the degree to which these organisms can have an effect. And you may remember in the mice that we described, which had an enormously penetrant deficiency because it was a null mutation. Those mice, of course, allowed access to the environment, but still the organisms that had gained access were very selective, three bugs. And we think that this is of importance, not just for colitis, which is what I've been talking about, but in fact for metabolic disease, for cancer, and a whole load of important and interesting things. So that we're very excited now to go on and develop this, and of course all other people will do that, and that's good. That's what science is all about. And so we think that this system really boils down to these four factors that affect disease. I'm finishing, right? Sorry. The microbiota I've talked mostly about, but they are themselves slaves of our diet because we feed them. Our genome, right, which determines to what degree we allow them access to our innermost parts. The inflammatory response, which then develops from that, and this is basically what is probably the key simple components of this system. So just to finish on this story, it all started off with these guys down here in red who got the original thing going. Iran Elinav, who now has his own lab at the Weizmann, Till Schrovig in Germany now, Jorge, who's at Penn, Chung, who works with Tyler Jacks. Now she was a grad student, she's now graduated, and Marta Zlodarska was the one who really pioneered the mucus story. In the beginning, I didn't really spend much time, and she was a grad student with Brett Findlay, has now moved on. And the story about IgA that I described was really these two marvelous postdocs who are still in the lab, Noah Palm and Marcel de Souter, and a lot of people that are tremendous help. Here we are.
Actually,